Okay, so welcome everybody to this new DaSCI seminar, the first one of this new academic year. Today is my great pleasure to introduce Dr. Adrien Bartoli. He has been a professor of computer science at Université Clermont-Ferrand since fall 2009 and a member of Institut Universitaire de France since 2016. He founded and leads the Endoscopy and Computer Vision Research Group, where, by the way, I did my first postdoc 11 years ago under his supervision. And he held an ERC Consolidator grant and an ERC Proof of Concept grant. He has received several awards, including the 2004 Grenoble EMP PhD Thesis Prize, the 2008 CNRS Médaille de Bronze, and the 2016 Research Prize from Université d'Auvergne. Adrien Bartoli's main research interests include image registration and shape from X for rigid and non-rigid scenarios, and machine learning within the field of theoretical and medical computer vision. In these fields, he has published approximately 100 scientific papers and has been on the program committees for top-ranking conferences. Today, Dr. Bartoli will talk about the research and industrialization of augmented reality systems for soft organs in abdominal minimal invasive surgery. I want to thank again Professor Bartoli for very kindly accepting our invitation, and whenever you are ready, the floor is all yours.
Thank you very much, Pablo. So let me start directly with this presentation. And you say the title. I suppose it may sound a bit obscure to some of you. So we're going to discover exactly what's underneath. So it's about a line of research we started about 15 years ago in Clermont, jointly between the hospital and the university and CNRS. I want to take you through this journey, so starting from a clinical problem that we define with surgeons to a product that we're now marketing based on the initial research that we conducted and the things we proposed through this journey. So we start with a clinical problem that we'll discuss very shortly, then the idea and the principle of the solution that we came up with. The main processing steps that are involved in this solution, then the test we made to be able to validate the proposed solution, the basic research it involved, the research software we developed, and finally the transfer and industrialization within the company. So that's the very short history of mini-invasive surgery. Traditionally surgery is performed by opening the cavities, the patients to see inside and to touch and cure and do something on the organs. But in the 90s came the mini-invasive surgery, where the patients were opened with very, very tiny incisions, and the surgeon would use a camera to look inside the cavity. So there was a huge advantage for the patients, but also difficulties for the surgeon because they couldn't touch directly the organs with their hands. In the 2000s came the robot-assisted surgery, where the surgeon holds a telemanipulator, and we believe that in the 2020s now are the era for digital surgery. So in this MIS, mini-invasive surgery, we have major advantages for the patient, faster recovery, improved outcomes, and also cost effectiveness, because typically the length of stay at the hospital is shorter. But there are critical issues because the hand-eye coordination for the surgeon is lost, the haptics is lost, and also the learning curve is much longer. Typically, the clinical problem we identified when we started working with surgeons are the following. So what you can see here are two different organs. On the left, you have a uterus. On the right, you have a liver. And typically, the surgeons, they do the surgery because they know there is something inside these organs that they have to remove or to fix in general. And they can see that on the scans they do preoperatively, typically CTs, MRI, and all these imaging. But when they do the surgery, they face the organ, which is not transparent. That's what they see with the cameras. And the main question is, where are the tumors that I have to remove? and where are the vessels that I have to preserve. So we came up with the idea that there was a visualization problem which was hindering the use of this mini-invasive surgery. So the question was, how can we help? How can we improve visualization to make this mini-invasive surgery safer, faster, and also easier for the surgeon? So from there, we worked our way and we developed an idea which was to modify the surgical image in real time during the surgery by adding information directly on this image. So that's what you can see on this example. So that's still a liver image. And what we've done to this liver, to this image, is to add information. So what you can see in yellow are two tumors which are inside the organ. And we're showing a sort of visual transparency that shows the surgeons where they have to cut to find these two tumors. And in blue, you have a part of the vessels that they shouldn't harm, of course. So that's called augmented reality. And the principle of augmented reality, as you can see, is you take a real image. Here it's a photo of the box on this example, a virtual model, which here is this Statue of Liberty, and you put this virtual model somewhere on the image, but with coherent interaction with the structure of the real world. And that's the difficult part. So in mini-invasive surgery, there are several ways to do that. We can consider an image and extract information directly from this image. For instance, with a neural net, we can extract the bleeding areas from this image and overlay this onto the image. This is a form of augmented reality where the virtual content is taken directly from the image itself. Another way is to use a second modality intraoperatively. So here it's laparoscopic ultrasound. You can see this black probe inside the image, which is an ultrasound probe entering the patient's cavity and also visible from the camera that gives an ultrasound image that we could overlay directly on the image with this green plane you can see that allows the surgeon to visualize directly the ultrasound in the image. And the last idea, which is the one we pursued, was to use preoperative imaging. So the imaging that is used before surgery for diagnosis. Here you can see a CT scan of the patient. In the CT scan, we can see the liver very well. We can also see the tumors, we can see the vessels. And the whole idea we wanted to achieve was to sort of merge the CT scan with the surgical image to obtain the augmented reality visualization. So we take the information from this diagnosis preoperative imaging of the patient. So that's a summary of the key idea. Typically, the patient goes into the scan for diagnosis, then the surgeon looks at the scan, and if the patient is scheduled for surgery, then of course the surgeon will have the scan in mind to navigate, but they'll be either on a different screen or just in his mind because they've looked at it before the surgery. So our key idea is here. We want to reintroduce this information directly on the screen of the surgeon during surgery by making this fusion. So concretely, we bring a PC to the operating theater, we plug it to the camera, and we have a computer program that reads the frame information from the surgery and modifies the frame before it's displayed to the surgeon. So locally, we started to work on that in the academic research team. It's called ENCOVE. 15 years ago, that's an academic team which is also partnered with the hospital. And 10 years later, in 2019, we transferred these results to a company that we started called Surgical Augmented Reality, Surger, which has made an industrial prototype. I will tell you more about how it went to transition from this academic product to an industrial product which can be marketed. So that's where we are. We've defined the clinical problem, the idea, and the principle of the solution. Now we want to see how we implement this computationally. So that's the setup. First, we have on the top left this CT scan, which is on the PAC server at the hospital, which we retrieve and store in this PC, which I was mentioning, which is in the operating theater. Then this PC is equipped with a capture card, which is connected to the camera and receives the images in real time and makes this fusion with a CT scan to obtain the image we want. So our challenge is the following. We want to implement software which combines preoperative and surgical images into augmented surgical images. So it's very difficult because we have two different modalities, right? The CT scan doesn't look like the image. They're very different. One is 3D, one is 2D, and they don't show the same intensity, the same color at all for the same structures. Then there are no fixed structures. There are no bones around the liver, which we can use as landmarks to know where we are exactly between the two modalities. And finally, there are huge deformations because of technical reasons, owing to this surgical procedure, but also because the patient changes position, because the disease might evolve between the scan and the surgery, etc. And of course, because the liver is also a very deformable organ, so we will have to deal with this. This is the plan with a bit more details on how we're solving it. So the first thing we do is to try and reduce the difference between the two modalities by constructing a 3D model from the CT scan. It's a known technique. We've used existing methods for doing that. We basically go through the slices of the CT scan, which is a 3D image. We segment each of the slices, and then we interpolate through the segmentation to reconstruct a mesh representing the organ, the outer shape, and the inner structures, here the vessels and the tumors. Then we calibrate the camera. Camera calibration is extremely important. It's okay if you don't know about it or if you don't want to understand it now. Just remember it's a mathematical model that says how we can associate a pixel in the image to a line of sight in space. So basically, it's the set of points in space that would project onto a particular pixel in the image, and it describes this relationship for the whole of the image. Then the most important step is called registration. It's basically the step where we bring this 3D preoperative mesh of the organ, reconstructed from CT, into the coordinate frame of the camera. So basically, we have to deform it. Like I explained, the liver is very deformable. So if we take this preoperative mesh and project it into the image, that's what you can see on the left. It doesn't project at the right location, of course, because the coordinate frames are different between the camera and the CT scan. Registration is, roughly speaking, to find this difference between these two coordinate frames so that the liver model will project at the correct location in the image. And the last step, once we have solved this registration, is to bring the information inside the liver onto the surgical image. It's called the augmentation or visualization. So we can achieve this with existing techniques. So for instance, for the first step, we have MITK, which is a free software to treat these medical images. Then for step 2, we have OpenCV. For step 3, we have ITK. And for step 4, we have OpenGL, for instance. So that do the job, all these to some extent, but it's largely perfectible. Actually, if we would use all these tools, it wouldn't be very precise and it wouldn't be very useful in the end. And the critical step, as you can imagine, is this third step of registration, because if we bring the liver model at the wrong location, we're going to show wrong information to the surgeon. So that can be extremely dangerous if the surgeon trusts the augmented reality system. So we have to be extremely careful with that. And that's why we had to conduct basic research to improve each of these steps to make them more accurate. That's the modeling we started from. So like I said, we take this CT scan and we transform it into a model. What is a 3D model? It's a collection of points. We typically call them vertices. Each point, each vertex has an XYZ coordinate in some coordinate system, intrinsically related to the CT. Then I was saying about camera calibration, which is basically the projection function. So given a 3D point x, y, z with camera calibration, we can predict the x, y in the image from this by projecting these 3D points. So given that, it's extremely tempting to say I have a 3D mesh, which are 3D points. I have my projection function from camera calibration. Let's project it. If we do that, of course, it falls at the wrong location in the image because they're in two coordinate frames which are different. And that's the registration step I was explaining. We want to take these XYZ coordinates of all these 3D points on the model and apply a transformation to bring them at the correct location in space in front of the camera so that the projection will act on them properly to bring them properly at the right location in the image. So the problem here is to change all XYZ to satisfy some constraints. These constraints, they are related to biomechanics, so we can deform the liver, but not arbitrarily. They're related to anatomical landmarks, which are very specific places we can identify on the liver in the CT scan and in the image that we want to bring together, and what we call visual cues, which are things from the theory of vision. So all of this is modeled by the theory of shape from template, which we have extensively worked on in this research group, and which was related to this problem of registering the liver and also the other organ we've been working on. So biomechanics, like I said, is the ability to deform in a non-arbitrarily way the liver. So if you imagine we cut the liver into very, very small pieces, each of these pieces, like in finite elements, can be moved but cannot be squished completely. That's a very sensible thing. The anatomical landmarks are these anatomical places we can find on the liver model and in the image that we want to bring together. I'm going to skip this detail because if you don't know, if you're not familiar with the anatomy of the liver, this might be a little obscure. Just remember it as a collection of places on the liver which we can identify in both modalities, hence that we can use as correspondences. There are quite few of them, and they're not actually points, they're curves, which make the problem a little bit more difficult than if they were points. And the visual cues, they are the typical things we use in computer vision. That would be the silhouette of the organ, and that would be things related to how the organ reflects light, which is very good in this type of with this type of cameras, these surgical cameras, because they hold a light source at the tip collocated with the camera, so we can model this extremely well. And modeling the light, the shading, which is how the diffuse reflection come out of the surface, and also the specular reflection is extremely useful to constrain the registration. So from this basic research, we developed a research software because there is the theory, of course, this modeling we've just discussed, but at some point we need to translate this into a computational software that we can use to make tests and in particular to characterize the accuracy of the augmentations we do, to test on animals, on patients, and see if we can really bring something to surgery. So let's see how we developed this software. So I'm coming back to the big general schematics I've shown with these 4 steps. For the first steps, we used MITK, the one software I mentioned already. But for steps 2, 3, and 4, we developed our own software, our own integrated software. Remember that step 1 is done before surgery, right? So we have this CT scan, we fetch it from the hospital, we prepare the model, and this is done before surgery. But steps 2, 3, and 4, they're done during surgery. So it's important that we have a software with a user interface that we can use conveniently inside the operating theater. So here we developed a piece of software in C++. We used CUDA to make computation faster, and it's all based on libraries, including OpenCV for the vision part processing, OpenGL for the rendering part, and Qt for the user interface. We also have a capture card in the PC and a GPU, of course, for the computations. If you're willing to know more about all this, we have papers describing these theory and the implementations available on the website of the research group and Cove. So this is how it looks in the operating theater. So here you have a surgeon doing a liver surgery on the patient who's here. This surgeon is a member of our research team, and you can see he's got two screens showing the regular view from the surgical camera. These two screens are on the right where he's looking at. And then on the left, you have the ultrasound screen, and leftmost, you have the view with our software running, showing augmented reality. On the right image, you can see an example of the user interface, which can be used to solve the registration, but also to edit the registration. Here you can see the liver model is superimposed on the image, and there is a sort of cage with control points that the surgeon can use if they want to edit basically the registration of the model onto the image. So that's typically how it goes. You have a 3D model, projects it. It doesn't project first at the correct location, and then it's deformed by the software automatically to fit these yellow and green and red curves that you can see. So the yellow curve is the silhouette of the organ, and the red curve is one of our landmarks. It's the lower ridge of the liver, which is a very sharp part of the liver that we can recognize in the CT and in the surgical image. I'm playing the video again to see for you to see, so you can see the inner tumors in yellow. They're following the motion constraint from the outer part of the organ, because we have this volumetric model with finite elements which cover the whole volume of the organ. This is another example. Now is the uterus, the one you've seen at the start of the presentation. And in these examples, we're superimposing a 3D model where we have shown two tumors in yellow, the outer part of the organ in white, and the womb of the uterus, and also the fallopian tubes in black. So with this type of visualization, of course, the surgeon can target directly for the tumors, but they also can spare the womb for future pregnancies, and also the fallopian tubes, which are extremely important for the patients. Here you can see this registration is updated as the video is going on, so as the surgeon is moving the camera or the organ, because the uterus is an organ that can be moved from the outside. To achieve that, we have tracked the organ in real time. And for that, we have developed a method which uses key points, and they're matching through the images. So we solve the registration on a few key images, and then we update it as the camera moves by tracking the organ. Another example that we've tried was on a partial nephrectomy. This one is done with a robot, a Da Vinci robot. You can see it's a typical view and tools that they use on this robot. Here you can recognize if you've seen this before. And the augmentations here shows the tumor, which was very close to critical structures of the kidney. And we've used the same tracking engine as for the uterus. The kidney is more or less rigid. It deforms much less than it's not entirely rigid, but deforms much less than the liver. The biggest problem with the kidney is the visibility, because it's usually buried in fat, and you can see a small part of it until they would remove all the fat, which they rarely do, except if they need it for a particular reason, but in general they don't. So it's a challenging case for these visibility issues. The liver can be seen very well. The uterus can be seen extremely well, and it's probably the easiest case for computer vision. But the liver and the kidney are very difficult for deformation and visibility, respectively. So that's where we stand. We made these research prototype, and then we embarked on the last step of the journey, which was to transfer that to a structure which would be able to make an industrial prototype. Of course, during our research, we made a user interface. We developed something we could use in the operating theater, but the surgeon was never alone to use this, because this was only test with us in the operating theater, handling the software. You know how it is in the academy. We're not research software developers, so we do make these user interfaces, but they're not really good for the users. They're more for us to be able to test things. In the industry, it's different. They're user oriented, and they want to develop a product which is designed for the end user directly. And here the end user is the surgeon, who's not a computer scientist, of course. So we embarked on this transfer 5 years ago, and very importantly, you'll see that not only should this transfer develop a usable software, but should also develop the software that can be CE marked. And CE marking is very important. It's whenever you make a medical device, you can only sell it if you have a CE marking given by a notified body of the EU. So we created a company called Sugar. Like I said, 4 years ago, it was myself with 3 surgeon colleagues, 2 gynecologist surgeon and 1 digestive surgeon who takes care in particular of the liver. And currently we have a structure with several departments, including administration, research, design and development, regulatory and quality, data science, and marketing and sales. We have 35 employees, and we spend a lot of time developing the software cleanly and following all the regulation rules, which allows us to obtain the CE marking eventually. So this is the big schematic that we followed for research, and we followed the same principle on the industrial software. We actually developed a suite of software. It's called X Sugar, and X is a generic letter that stands for the organ which is to be addressed. So U sugar for the uterus, L for the liver, and K for the kidney. And the first step is solved by a software called Sugar Plan, where you can edit your segmentation. You can segment your preoperative image, edit the segmentation, edit the model, and export it for the surgery. We're also compatible with external software, commercial ones, because there are many which are on the market already to construct these 3D models. They're called digital twins, and they're actually very trendy. And for us, it's very good because it's one of the crucial steps of this workflow, and it means that there is software out there that can solve this step very efficiently for us. We just have to make sure that we can read their outputs, the outputs they export as models. So we've developed these 3 softwares to address the 3 organs, and we've also developed a platform which is in the operating theater. It's called a Sugar platform, which also has to be certified to be marketed and to be used for real surgery. So this platform is this following design. So we have a card, we have a computer with a capture card to collect the video of the surgical camera. There's a GPU for computation, and there's a tactile screen with which the staff can interact to set up the software and to make it run and to customize the different options. Everything is on a card that can move in the operating theater. It's a class 1 medical device, so it's the softest class, the easiest one to pass. And then we have on this platform the 3 software I was mentioning, so the uterus, the liver, and the kidney. These software are class 2A medical devices, so it's a more restricted class. So for them, obtaining a CE marking is more difficult. So we started the company in 2019, and we were more or less done for the platform in 2022, so 3 years after. And now the certification is done for this platform. So it's we can officially bring it to operating theater and use it in the operating theater. For the software on the uterus, which historically was the first one that we started to work on, even for the research, the development was done in 2022, and all the time since then was used for the CE marking and the clinical study. Now we've obtained this CE marking very recently, actually in September this year, and we're going to proceed to sales very soon. For the liver software, the development is ongoing, and we're planning to have a CE marking by the end of 2025. And for the kidney, eventually, we're planning to have a CE marking at the end of 2025 or 2026. So I'm going to skip this part because I want to save it for the question if there's any on the industrialization process. I want to show you some results. So it's the ongoing clinical trial we're doing on the uterus software. So you can see the platform and the software in action at the in the operating theater. The main endpoint of this clinical trial is to see how the software can ease basically the workload of the surgeon and reduce the stress during the surgery. The secondary endpoints were the risks and the feasibility of setting up the whole thing in the OR, and also the impact on the clinical outcomes. So far, we have including 29 patients. The study is planned for 40 patients, so we're very confident that we'll reach that. So on the screen, you can see the augmented visualizations. Here, there's a very big tumor. You can see it in green. It's a myoma inside the uterus. And we have 3 participating hospitals for this study. So we've started inclusions of patients in 2 of these centers, and we're looking forward to the opening the 3rd center very soon. So, sorry, I skipped one slide, and I'm going to skip back to it. Yes, if I can. So, yes. So that's the current version of our software for the liver. On the left video, you can see the preoperative model of the liver, which we collected from an external software, Synapse 3D by Fujifilm. In the middle, you can see the intraoperative software, the one which is used during surgery, where the model is overlaid on several static images of the liver. So there's a big difference between the uterus and the liver. Like I said, the liver is extremely deformable. So as of today, we don't know how to track the liver reliably. So we just augment fixed images, which nonetheless is a very valuable help for the surgeon to navigate and to find where the tumors are. In our user interface, the surgeon can click on the model directly during surgery and define his resection plan. So for instance, if they want to enter this way or that way into the liver, they can see the trajectory of their tools to reach the tumor, and they can see this both on the 3D model and thanks to the registration on the surgical images on the same screen. And this is the software we've developed for the kidney. These pop-ups you see are the software capturing keyframes to improve tracking automatically. So don't pay attention to it because I haven't described exactly how this algorithm works, but you can think of it as the software being able to register new images as they come, showing new parts of the organ as they're discovered. Here you can see it works in real time, and the augmentation is actually quite stable thanks to this keyframe mechanism. There are two images side by side because here the camera is actually a pair of two cameras. It's a stereo camera because it's the one from the robot where the surgeon has a 3D visualization thanks to the headset in the surgical console. You can see it's tracking well, and in this case we were extremely successful. It was tracking the kidney for a large part of the surgery, so you can see the cameras going outside of the patient cavity and then back inside. Perhaps it was outside for cleaning or something, then back inside to continue surgery, and the software is able to relocate automatically the organ and to re-register automatically the preoperative model to achieve augmented reality. Another short video, short teaser for the kidney software. You can see here a surgery where we have customized the visualization mode so we can show certain anatomical structures. We can show the vessels, we can show some of the ducts of the kidney, and also, of course, the tumors, which is the important part for resection. Launching the other videos where you can see examples. The bottom right one is interesting because it wasn't a kidney surgery per se, it was a thrombus surgery where the vein outgoing from the kidney was actually contaminated by a tumor, which was blocking it. And we used augmented reality to locate this tumor within this vein so that the surgeon could see it. So it's in this, that's basically the robot's vision you have here on the whole screen. And in the bottom right, what you have is the augmented reality view with the tumor in green. So at Sugar, we also do some data science to feed all these software. They're used for two primary purposes. One is to be able to locate what we call the landmarks. So I was describing it when I explained the registration method. It's basically these places on the organs that we can use as correspondences between the 3D model and the image. And of course, we want to avoid the surgeon having to mark them in the surgical image by hand, because first their hands are in sterile gloves, so they can't touch a mouse, of course, and also it takes time and disrupt from the main task of surgery. So we're developing neural nets to detect automatically these landmarks. And what you can see on the right are initial results on detecting some pathologies. Here it's endometriosis. And the red parts are the areas where the surgeon has to do resection, and the green parts are where these areas that they should check very carefully. It's surprising, but it works extremely well according to the surgeon feedbacks on these results. So it's the two basically types of data science we do. So we have a whole process for making contracts with the hospitals to gather data, for collecting, anonymizing, storing, and annotating. So everything in the company has to respect the constraints designed for CE marking later on. If we fail one of these steps, the notified body will notice, and we can't get the CE marking embarking any of these tools anyway. So we developed a recorder that we can bring to the surgical rooms, and a whole pipeline where we can view the videos, export them and share them, and also upload them on the supervised lead platform for annotation. So that's the end of this journey. We started from the clinical problem and we ended up with a product. This product is going to be sold soon. Bear in mind, it's a 15 years journey from the initial discussion with the surgeons to the end product. So it was a very long time. And what you can see in this diagram is that this is not a linear process. This is not a fixed process. There are overlaps, there are failures. They are going back and forth deceptions and great and very bad moments as well. It's all but linear. So whatever one does to do translational research from the academia to a product will not necessarily follow these steps. This is just an example of how it can be. And in practice, it's of course more complex than that with all these non-linearities. So thanks for listening, and I would be very happy to take your questions.
So thank you very much, Adrien. Well, now it's time for questions from the audience. So as usual, feel free to write your name in the chat and I'll give you the turn to speak. Any questions? Yeah, well, Edgar, please go ahead. Yeah, Edgar, you can ask your question. I don't know. Well, in any case, Javi Melero, if you want to ask your question.
Hello? Hello?
Yeah,
Hello,
great.
do you hear me?
Yes.
Yes.
Yeah, Edgar.
Okay, thank you. Thank you very
Yeah.
much. First of
Yeah, go
all,
ahead.
I'd like to congratulate Adrien Bartoli for this brilliant presentation. And I'm wondering about the challenges, the possible challenges you have encountered when you implement the software. If you should discuss a little bit more about the biggest challenge maybe you have found to implement.
Okay, sure. Are you referring to the research implementation we've done at the university or the industrial implementation we've done in the company?
The industrial, because I suppose that you had to follow some steps, some policies, and some constraints in order
Yes.
to implement the industrialization.
Yes. So let me come back to this slide. Probably, I don't know if it helps, but in terms of software development in a company, everything we do in a company, actually, not just the development, has to follow protocols which have to be written in accordance with norms, regulative norms, and failure to comply with this, of course, will cause failure to obtain CE marking. So the first thing is to follow these norms, and by following these norms, you can have a certificate that you're a medical device manufacturer, software manufacturer. That's the first thing. And that's mandatory for the CE marking. Concretely, it means that when you have a developer creating code, you have to have another developer reviewing this code and giving feedback. And there's these constraints, so it's never one developer on his own to make code, but there's at least 2 or 3 around to review the code. So there is all this code review, and also we have to have unit tests and follow a methodology for this development which is compatible with the protocols. So I can't tell you much more about it. It's not my field in a company. I'm not doing the development, I'm doing the research, but I know it's extremely constraining, and we have to be extremely strict about it.
Okay, thank you.
Okay, thank you. So now, Javier, whenever you want.
Thank you very much, Adrien. It was a pleasure to see your speech and to enjoy it, mostly because it covers several of my fields of interest: computer graphics, entrepreneurship, artificial intelligence, and many others. I have a couple of questions, one related to computer graphics and the other to the entrepreneurship. One of them is, how do you handle because you have answered, how do you handle the non-linear deformation of the surface, which was one of the questions I was making myself, but you explained it later. But what happens when the organ is broken, is cut, and is non-linearly but not rigid and not elastic, deformed either. It's completely changing the shape. So I don't know how do you handle it currently, because it is an alignment or a registration program quite hard. Do you also simulate the cuts in the organ in the digital twin, or it is a step still to be done? And the other question regarding entrepreneurship, if I didn't understood properly, you told that you were 45 people in the company?
55.
And I mean, research in medical devices is expensive, but entrepreneurship in medical companies it's even more expensive. I would like to know, if possible, how much and who funds all the investment to be done, because this will not be solved until 1, 2, or even 3 or more years, and it is not an easy. There is not an easy channel to reach, to recover, to receive all the money that has been invested. What's your plan for that? I have 2 questions, and I take the opportunity to recommend all the students that are here to think on the future after the PhD and think on how to develop their own company. Thank you.
Well, thanks for your questions. They're very interesting. I mean, the registration with cutting is extremely difficult, and you've probably noticed that I didn't show any example with cutting, right?
A small one in the kidney.
Yeah, exactly. But it wasn't a huge change of the shape. So you've asked the right question. So as we stand for the company's product, we don't have any handling of cutting. As long as the organ shape doesn't change too much, it works well. But we don't have any special mechanism to deal with the cutting. We have tried something in the research group at the university, which is basically to detect the cut in the surgical image, and we assume that at this point we have a registration which is okay when the cut starts, and we sort of transfer the cut onto the 3D model. So we're going the other way around, and we basically break the edges between the vertices along the cut path. So we sort of update the 3D model, and then we recompute the registration.
Okay, good. And
so this works, but this is unstable so far. That's why we haven't included it in the commercial products yet.
Okay.
And the other question about the funding, essentially, is you're absolutely right. It takes a lot of time and a lot of money as well. So the company is out there for 5 years. Actually, it was created in October 2019, so it's exactly 5 years. And only now we've obtained the CE mark. So it means so far we haven't made any single euro. And we've lived on investments, and these investments, they're mostly private, from private investors, corporate investors, and also business angels. It takes quite a lot of our time to deal with this, quite a lot. We've done 2 fundraising, the seed one initially, and now we've just signed for the Series A in July. That's a huge part of the work being in a company, to make sure these fundraising happen in time, at the right time, and also to find investors interested in the project. Yeah.
And how much? Sorry, the last one. How much of the company do the researchers hold? How much? How large is the percentage of the researchers, of the founders?
For the co-founders, I can't tell you exactly, because it's changed with the Series A fundraising. So I wouldn't know exactly
roughly
today. Not even roughly. So we have our
10,
first
15?
board meeting tomorrow, and I think that I'm going to understand things more clearly by then.
Thank you.
Thank you very much. Any other question from the audience? Question? Well, I can ask you something, Adrien, because well, I have two related questions that I guess in the end represent the same one. The first one will be, I mean, how do you evaluate this system? What kind of ground truth or objective evaluation metrics do you have to evaluate the robustness and precision of this registration approach? And related with this, what kind of key performance indicators do you have in order to know that your system works sufficiently well and it's going to pass all this healthcare legislation? Because I guess that since we are talking about healthcare, there are many performance, safety, and bureaucratic milestones that you have to meet.
Yeah, the first requirement they have is that the system works, so it's not going to stop in the middle of surgery. So it's not evaluating the performance per se in terms of accuracy. It's evaluating whether it works at all reliably. And the second one is an interesting one, which is very usual in research, which is the TRE. So this target registration error on the structures that we're predicting in the organ. So typically the tumor and how accurate we're predicting its location, which of course we cannot evaluate easily on patients because by definition we can't easily make a measurement. Otherwise we wouldn't need augmented reality at all. So we have models with animals, with phantoms. So we basically print organs in 3D that we reconstruct from patient scans, things like that. And with that, we have full control of what's going on. So we can measure this way. That's how we made it through the CE marking.
Okay, great. And well, from my point of view, if there's no more questions from the audience, my last question will be, from the scientific point of view, in all these 15 years of research, from your point of view, what has been the most challenging part? If there's something that comes to your mind to say, okay, the most difficult research part in all this pipeline was this.
Well, I think it's the modeling of the complete constraints we have identified, and maybe this is probably not the final word. Of course, there might be others better suited to this problem, but modeling the light, modeling the camera, modeling the reflection, and also identifying the anatomical landmarks was also a challenge because it requires talking to the physicians and understanding each other. And that's quite a challenge because it's really difficult to read a CT scan when you're not a physician, and also if you want to identify what you can see in the CT scan and in the image, it requires a lot of getting used to this type of surgery and to the anatomy of the organ. So for me personally, that was a challenge and also a huge interest.
Okay. Well, thank you very much. I don't know if there's any other question, the last chance for the audience. No? Well, so I think we can end here this DaSCI seminar. I want to thank you, Adrien, again for accepting our invitation and for giving
much.
extremely interesting talk. Thank you.
Thanks.